Welcome to the basic structure of a cell. This is basically just a review of what you learned in freshman biology. So we're going to skim the surface pretty quickly and move on to tissues after this unit. There are three basic types of cells. There's the eukaryotic animal cell, which is what we deal with mainly because we are human. There's the plant cell, and there's the bacterial cell, which is also called prokaryote. Okay, the cell theory is that all living things are composed of at least a cell or more. But the cell is the basic unit of structure and function in an organism, and all cells come from pre-existing cells. Okay, prokaryotes, again, is the big fancy name for bacteria. They do not have a nuclear membrane. They don't have a nucleus. They basically just have all their DNA floating inside their cytoplasm. The eukaryotic cell is us. U means true. We have a nucleus. Let's see. The nucleus and the cheek cells up here, a cell membrane which contains everything inside the cell, and then cytoplasm. Cyto is the root word meaning cell, plasm is that gooey squishy stuff. And organelles are the tiny little cell organs or parts of the cell that function like organs. Alright, again, cell size and shape is determined mainly by the ratio of the outer surface area and the volume. A cell, because it is membrane bound by a phospholipid bilayer, it can't get too big, otherwise it'll explode basically, it'll fall apart, it'll pop. So surface area and volume. There's the phospholipid bilayer. There's a nice little diagram of my cell. So the phospholipid bilayer, it encloses the whole cell. It's made of a hydrophobic and hydrophilic components. And very important, it is semi-permeable. That means it lets certain things flow on through, other things need to be carried through. It's a fluid-like, makes it squishy, not stiff, like a plant cell. So hydro is water, phobic is fear, philic is love. So the hydrophilic phosphate heads stick outwards towards the outside of the cell and the inside of the cell, the cytoplasm. The hydrophobic tails are made of little fatty acids, lipids. So that is, they're fat, basically they're oils. They abhor water, so they face towards each other and stay away from water. Again, it is a phospholipid bilayer, the phosphate heads, the lipid tails, and there's two layers, as seen here. One layer, two layer. They do receive materials through passive transport. That means things can pass right through the protein channels, and active transport, which requires ATP, and membrane-linked enzymes and receptors help. You got sterols that help stabilize the lipid bilayer, make it a little more firm. And here's a beautiful little diagram, artistic rendition of the phospholipid bilayer with the antigens outside. You got some oligosaccharide, which is many sugars. You got a glycoprotein, which is a protein with a sugar attached. You got these proteins, which are called porins. They let things through. And there's your lipid core, your phosphate heads, phospholipid. Again, porin is a special type of protein which acts as a gate, basically, in the cell membrane, allowing certain molecules in. Porin ends with IN. Protein ends with IN. Most proteins end in IN. Not all, but most. Alright, active transport requires ATP in order to allow material through the cell. Facilitated diffusion has a carrier channel which allows only certain things to go through the channel. So, kind of like in a canal, you might have a thin little gate which allows rowboats through and kayaks, but not bigger ships. And of course, water diffuses by osmosis. Osmosis refers only to water. So there's a closer view of the phospholipid. You got your phosphate head, and you got your fatty acid tails. There's two fatty acids to make the tail. So the organelles are very small. You can only see them under a microscope, and they all have their separate functions like the organs in your body. So the endoplasmic reticulum, endo means inside, plasmic, plasma. Reticulum is folded. So you've got these folded canals for movement. You got your Golgi body, which wraps and exports proteins. It modifies it. Nucleolus is within the nucleus, and it produces the ribosomes. Lysosome, a zome is a vesicle basically. Lysosome is a vesicle which contains lytic enzymes, or enzymes which break things up. And it gets rid of, it's kind of like someone in their presentation said, it's like the stomach of the cell. Digests it, and then it gets rid of the waste. And then ribosomes, first word in ribosomes is ribs. When you order ribs, you're really not eating the bones, you're eating the muscle, which is protein. Alright, the Golgi apparatus, or Golgi body, is a flattened sac. It just looks like a flattened sac stuck there. And it has a receiving side near the ER and smooth ER and rough ER. It takes in what they've made. It then modifies it. It can take the proteins and add some lipids to it, some sugars, and make a phosphoglycopeptidolipid of some sort. It can make take the hormone, package it up, and then its membrane is the exact same type of membrane as the cell membrane, because those vesicles just move outwards, connect to the cell membrane, open up, release their contents, and become part of the cell membrane. Why does the cell get bigger? Because the cell also engulfs things, which I'm going to talk about later, so part of that membrane comes back in terms of vesicles. So transport vesicle is just part of the cell membrane. So a lysosome, as I said earlier, lytic means to chop, to break, contains digestive enzymes that breaks down food and worn out cell parts for the cell. So it recycles itself. It is programmed for cell death. Cells get old, they eventually break down from within, basically. Your cells are always dying and being replaced, for the most part. Nucleolus, again, you can have 1 to 3 nucleoli, plural of the nucleolus, inside the nucleus. During mitosis, they disappear. And during interphase, they're visible again. They do make ribosomes. They produce the ribosomes that make the proteins. The nucleus is the control center, it's the library. It contains all the different menus for every different little thing your body produces. Different cells have different parts of the DNA read. Centrioles, these two little things, appear during mitosis. They line up at the end. They pull the spindle fibers, which allow all the chromatids to pull along and separate from one side of the cell to the other when the cell undergoes division. Smooth ER is smooth because it hath no ribosomes. It basically works with the synthesis of lipids, fats, and steroids. It does concentrate calcium, and it helps drug detoxification. So it's kind of like the liver of the cell as well. It helps detoxify and metabolizes steroids. So it's much like the liver. And since it's smooth and fats are smooth, you should be easy to remember that it is the site of lipid synthesis within the cell. And it's, like I say, a manufacturing plant. Number 4, over here would be your smooth ER. Number 3 is the rough ER. Has ribosomes on it. And this big glob over here in the pink would be your Golgi body. Can you see how the smooth ER is producing stuff which comes out here to the Golgi? Is being packaged in the receiving end, modified, transferred on the sending end. Number 10 goes off the cell membrane. Alright, the rough ER again is rough due to ribosomes. Protein synthesis, ribs. Reticulum means reticulated or highly folded. Proteins are made up of amino acids. Remember the building blocks. This is a freshman biology, basics. Amino acids link end to end, make polypeptides which become proteins. Lipids are made of fatty acids and glycerol. Carbohydrates are your sugars and starches. Carbohydrate of 1 to 2 to 1 ratio, or 6 to 12 to 6, which is a 1 to 2 to 1 ratio. Carbon, hydrogen, oxygen, which would be carbo, and then H2O, hydrate. Nucleic acids are your DNA and RNA, deoxyribonucleic acid and ribonucleic acid. They both are made up of a nucleotide, nucleotides, and a phosphate. The mighty mitochondria. It is the site of respiration where ATP is recharged from ADP and AMP. ATP is adenosine triphosphate. Those 3 phosphate bonds are where the energy is stored. So when you lose energy, it becomes ADP, adenosine diphosphate, which still has some energy in a phosphate bond. When that phosphate bond is broken, it becomes cyclic adenosine monophosphate. With only 1 phosphate, it now becomes cyclic. And that little tiny C, the C stands for cyclic, because it forms a little circle, becomes the hunger signal for the brain. It travels through the blood, and it fits right in a receptor in the brain, which says, I'm hungry. That's how you know you're low on energy and you're hungry. Cytoplasm, cytocell, plasma fluid, we discussed this once. Again, brief review. Cytoskeleton, it's like the skeleton of the cell. It's made of microtubules and microfilaments, little tiny protein tubes and filaments which form a scaffold across the inside of the cell. Microtubules help support, microfilaments help support and allow for some transport within. So along these filaments, kind of like a zip line, stuff can zip across from one end of the cell to the other. Cilia are, that's a horrible diagram over there, but cilia basically extend on the surface of the cell. We have them in our nasal passage in our throat, moving the mucus constantly and daily back down into our stomach. When it gets really cold and these cilia slow down, the mucus doesn't get shoved back as much, so the mucus being produced starts running down our nose, and that's why we call it a runny nose, because the cilia are too slow now when it's cold to move them back. Flagellum. The flagella is an amazing thing. If you look at the black and white picture, it shows an artistic rendition of how these proteins line up within the cell membrane. The flagella is mainly used in sperm for locomotion during reproduction. Endocytosis. Endo is into, cyto again is cell. Any type of thing in the cell. If the cell invaginates or folds in and takes in water, that's penocytosis. Pinao is Greek for drinking. So help think of it as a pina colada, drink. Not that the cell's getting drunk or anything, but pino is drinking. Phageo is Greek for eating, phageo. So phagocytosis is cell eating. Anytime the cell invaginates or wraps its membrane around something and pinches off and takes it in, that is phagocytosis. It could be a white blood cell attacking a bacteria. It could be a regular cell just taking in a solid molecule. And here's a nice little diagram of phagocytosis of cholesterol. You might want to pause it here and look at it. I'm going to move on. So, which molecule would most likely enter the cell by passive diffusion? There's your answer. It's basically lipids, because you've got a bilipid, phospholipid membrane, and water doesn't pass easily through lipids. Now, you've got some pores that let water through, but the starches are too big, proteins are too big, carbohydrates are too big, the big ones. Glycoproteins and polysaccharides are way too big. But lipids will transfer through. That's why a lot of the patches nowadays with drugs are a lipid-based patch. Alcohol and lipid-based, they can dissolve right through the skin. So know your cell, love your organelles, study hard for the quiz,